All right. Thank you very much for this opportunity to present and for everyone for staying to the end. I think this is a very special last session. The subject, if you think of it, is quite different. Rejuvenation is a very particular dream. We talk a lot about extending healthy life, therapies to prolong life, but rejuvenation is a dream of essentially life infinite, of immortality. If there is a way to undo the damage and keep undoing the damage, that's quite a different category, I would say, altogether. And I would like to stress something very special about our accomplishments in biomedicine of human history. If you think about the greatest accomplishments, I don't know what's your list. My would be painkillers. Right? It's an amazing miracle. Your head hurts, you pop a pill and it's gone. A miracle, really. Antibiotics. You treat a wound and it heals literally in front of you. Vaccines. Most recent miracle is genome editing. There is something in common. None of those we really invented, in the sense that we invented a bicycle or a jet propulsion engine. Those were invented by nature or evolution or whatever you want to call it. We observed, we understood, and learned from nature, often without really understanding how this process works. We move this into a new context. And so I think that rejuvenation will very much work the same way, and my talk today is about learning rejuvenation which already exists in nature, in germline. So I would like to start with acknowledgement. I'm talking about early stages of a project that was funded for 2 years generously by Amaranth Foundation. If you haven't seen their manifesto, Bottlenecks of Aging, I think all of you should follow the QR code and read. Those are important, underappreciated directions in the field. And I was fortunate to follow priority number 1, deciphering mechanism of germline immortality. So what are we talking about? What is this germline immortality? This is the point where I am missing my opera voice to sing. We do it, frogs do it, educated water fleas do it. Every single species which exists on Earth has a reset when they create the next generation. So if you think of it, at this point, we take 2 cells. I will not call those cells old, but those cells carry the insults of aging. Those were sitting in the bodies, not protected from ionizing radiation by some kind of a lead chamber, metabolizing, accumulating protein aggregates, affected by reactive oxygen species, and you mix those 2 cells, poof, the new baby is born young. It is so obvious that we think it's a really stupid question to ask. How come the new babies are born young? And conveniently, as I said, because every species does this, whatever secret you learn by studying this phenomena in a model organism should be a lesson transferable into humans. So the dream here is that if we understand what's going on, we could actually create therapies. There is supposedly a mechanism of quality control, a mechanism which undoes the insults of aging, which could be translated into old somatic cells, maybe. All right, so what is this damage we're talking about? It is suggested in the literature that oocytes, going through oocyte maturation, do away with protein aggregates. They sort deficient mitochondria to only send nice, healthy mitochondria, and they get rid of extra chromosomal DNA. So very important clue came when cloning was discovered. So this is Sir John Gurdon, who unfortunately very recently passed away and got a Nobel Prize for discovery of cloning. I remind you briefly that he has done this in frogs, in Xenopus frogs, and there's a very good reason, because Xenopus egg oocyte is a large 1 millimeter diameter ball. It's nice to manipulate this. It's single cell. And so the way that this worked is that you take a nucleus out of somatic cell. So you could start with an old cell, a cell from an old organism. You pop this nucleus into the oocyte, and the result is a cloned animal. Now, this germline rejuvenation is often discussed in the context of a rejuvenation of the genome. Right? Particularly in this field, there's discussion of rejuvenating with Yamanaka factors, for example, with resetting the methylation, and so on and so forth. And so those stories are relevant and interesting, but focused in the nucleus. Here, the evidence that the rejuvenation might be in the nucleus as well, but there is an important part that takes place in cytoplasm. And so this is what we study, what takes place in the cytoplasm. Something that is seldom discussed is that the cloned organism, not in the case of Dolly the sheep, unfortunately, but we now have cloned organisms in many, many species, and those organisms created by cloning have natural lifespan. So whatever happens here also carries the magic with it. So this is a paper from Science magazine, which suggests, from the lab of Cynthia Kenyon, suggests and provides evidence done by imaging that immature oocyte is, so to speak, full of garbage, that there is aggregated protein. And then somehow, going through the process of maturation, poof, this garbage is gone. So there is evidence. Now, in this paper, which was done in nematodes, there is a little noticed chapter in frogs. The mechanism which they suggest, they also observe as being active in frog oocytes. And so this is one reason to go into frogs, but there are many more. This is my own, now scary to think, 10-year-old work, where we have done quantitative proteomics of developing frog embryos. So what we've done is we collected time points of frog embryos developing from a single cell into a living, swimming, metabolizing, talking organism, which has many, you guys are hard to crack jokes with. So there are many complex differentiated tissues, all right? So this process takes dozens of hours, and what we were able to do by measuring RNA and protein in parallel and solving a simple system of equations is to calculate how many molecules of protein were deposited in the oocyte, Okay, so think about it. I take this organism, and all the work in the organism is not done by RNA, it's done by protein. I go and I pick a molecule of protein and say, where did this molecule come from? It was either deposited in the oocyte or synthesized during the development. And turns out that most of those were deposited in the oocyte, which was quite a surprise. And this is not a proof, but it's an evidence that you better deposit functional good protein and have quality control when you do that. Another odd reason that it was convenient to work in frog is that over the years I accumulated a collection of old frogs, perhaps the only collection in the world, including so-called space frogs. About 40 years ago, NASA wanted to know if vertebrate fertilization would work in the orbit. This was done by in vitro fertilization with frog oocytes and sperm in space, and frogs which were used as controls animals back then, which are now almost 40 years old, were around. Anyway, I accumulated a set of frogs with birth certificates, so we have material to study. But also, this paper just came out. We were able to develop and publish methylation clocks. So now if we go into the forest and catch a frog, we could actually attribute age to this frog and study what happens in the old ovary with old oocytes and so on and so forth. So I'm sorry if this is a graphic image for you. To me, when you open up a frog, you pretty much find everything. Okay, there is no prostate and no mammary gland, but all of the important organs and tissues are there. These are different stage of maturation of oocytes, where mature oocyte is about 1 millimeter ball, as I said. What's important is that it has enough material to do quantitative proteomics in a single cell. You could not possibly do the sort of manipulation and measurement with human oocytes, mouse oocytes. Pretty much frog is the only system where you could have thousands of oocytes going in parallel in synchrony and do quantitative proteomics and phosphoproteomics to understand what happens. Another important feature of frogs is that as oocytes mature, transcription stops. So at this point, there's no more transcription. So we in the biology love to measure gene expression, but here we could completely rule out any support by gene expression. Whatever happens, happens because there are changes at the level of proteins and post-translational modifications. So that's quite important. And so, as I like to say, the magic of quality control and rejuvenation happens here. Going from mature oocyte to progesterone stimulated oocyte. So this, you could call this ovulation. And observing what's going on there would supposedly give us clues. Now, you can imagine using this as an assay. If this system, and this is a system which undergoes meiotic cell cycle transition. Meiotic cell cycle transition only happens in our lives once at this point. Right? And we think that there is some activity, some enzymatic activity, which is able to chew through aggregated protein. If you identify how the hell it happens, you could imagine going into other harmful aggregated protein and activating this mechanism. So you could do all sort of activity assays by injecting aggregated protein into such oocyte and so on and so forth, interacting with drugs. And so this is a very simple experiment where you add progesterone to oocytes in the dish, you collect time points, and measure time series. You ask what changes, how do proteins change in the system. So this is what results look like. Those are just time series where proteins are grouped by dynamics, right? So here 500 or so proteins where nothing changes. It just 4 hours it stays flat. And what you see from this mosaic is that largely there's no change. Yes, there are a few well-characterized changes, but basically not much is happening at the level of protein synthesis and degradation. Now, here's the same organization of data with phosphoproteomics, right? So this changes of phosphorylation, phosphosites, and protein. And what I hope, jumps out at you is this dramatic difference. Everything changes. Phosphoproteomics is very dynamic. Now, if you at all think about proteins and post-translational modifications in your work, you would have heard of Phosphosite Plus. This is one repository of everything that we know about post-translational modifications of proteins. And transitions in meiosis was largely missing from this repository because it is very hard to do phosphoproteomics of meiotic transition. So essentially with this experiment we put on the map thousands of phosphosites and the dynamics. This is just a quick example for biologists in the audience who know and love cell cycle. CDK and associated proteins are probably the best characterized proteins. So here are some protein pathways where each protein is decorated by phosphosites. Whatever is in yellow are the phosphosites which we put on the map, which were not characterized and not known before. Right, so that's kind of a useful addition to encyclopedia of phosphoproteome. And I just want to stress one particular group of results, which has to do with nuclear pore complex. So this is perhaps the most conserved complex in evolution, and we see very active remodeling of nuclear pore complex. Now, here's just one of the protein in this highly complex assembly. Many phosphosites changing dynamically, and then a focus on only one of these trajectories where we give absolute stoichiometry with confidence intervals. So I'd like to take just a moment to explain why is this important, right? So what we detect are changes in phosphorylation of some protein. And usually when phosphoproteomics is done, it is done relatively. So you could say, well, there is this protein and this site on this protein, and it changes tenfold. Sounds important, but perhaps what happens is that the total level of phosphorylation changes from 0.1% to 1%. So 99% of all of the proteins are untouched. So the level of phosphorylation changed tenfold, but how important could this be? As opposed to tenfold going from 10% to 100%. Right? So the reason that we were able to get this level of information is our previous work, where we established the way to measure confidence intervals in quantitative proteomics, but also to compute, again, solving a system of equations, stoichiometry of turnover in this system of different phosphoforms. This dramatic remodeling in nuclear pore has come up previously. This is a work from a group at Berkeley by Elchin Unal. They study yeast, they study meiotic transition in yeast, and simply speaking, what they see is that when yeast undergoes meiotic transition, there are 4 new nuclei and then a compartment with aggregated protein. Essentially, yeast in meiotic transition packs the garbage into a special compartment which disappears. Again, this is imaging-based work, so the hope is that our work with quantitative proteomics gives a clue of how this happens. There's another group which does very similar work in mouse oocytes, where again there's evidence that oocytes going through maturation are packing together their garbage and getting rid of it. So we published these early results. The early stages of this work were quite successful, but there's much, much more to do. And unfortunately, I must say that the work have been stopped. About half a year ago, funding was from NIH to Harvard has been terminated. So if this sounds exciting, I would very much appreciate help. I would either like to have funding to continue this work at Harvard or an opportunity to continue this work elsewhere. But I'm quite excited about our results, opportunity to share with you. And with this, thank you, and I'll take questions. I guess I forgot to say, so I'm working with somebody who creates movies about science and wants to create a series about longevity field, which is semi-animated. So this was supposed to be, me on the quest for longevity.
So what information did the frogs give that other model organisms can't give?
We cannot measure proteome and phosphoproteome in tiny cells, right? So it's the features of frogs, not just having huge cells, but also the fact that transcription is stopped and other peculiarities of this organism allowed us to essentially create this map. Now, if you want me to tell you what to do to rejuvenate your body today, I'm afraid we will have to wait with this. Right, but we lay the foundation to understand what's going on, to compare to imaging work in other organisms, go back to changes in proteomics and phosphoproteomics. So that's the way biology works. We get clues.
So basically, since it is very ancient invention, this reset of youth, it should be the mechanism should be the same from yeasts to humans. So it doesn't matter on what organism to study. Am I getting it correctly?
Yeah, exactly. I think I pointed this out at the beginning that we should, from yeast to frogs to humans, the mechanism should be the same. There is some kind of a protein degradation regime that is activated there, which apparently is able to sort garbage from good proteins, not just bulldoze through everything. And so it's just not clear what kind of degradation. There are some clues and speculations by other groups, but exactly, we should be able to borrow the secret from nature.
Thank you very much.